So translation is part 2. And this is where the RNA will leave the nucleus and go to the ribosome to become a protein. So what happens is, this is going to occur in the cytoplasm. It requires all 3 types of RNA. That's going to be the mRNA that we just made, transfer RNA, which we'll just talk about in a second, and ribosomal RNA, which is where we're going to put this all together. And how I think about how these things are, and I'll sort of refer to them in a minute, is the mRNA is the recipe. And it's going to tell us how to put that protein together. The tRNAs are like chefs that are going to get the ingredients and put them in the correct order. And then finally, the ribosome is kind of the kitchen, the location where all of this happens. And the mRNA is read from 5' to 3'. And we're going to go ahead and do this all outside the nucleus. Remember that. It's going to be in the rough ER. So we've already talked about some parts of this. We've already talked about how DNA gets transcribed into RNA. Now we're going to cover this part, RNA getting translated into a protein. So that's going to be a new language. It's going to be a language of amino acids, where before we were always just going from one nucleotide to another. Before we get there, let's kind of discuss the players involved. You know that there was a tRNA, and the tRNA basically had a couple of pieces to it. Number one, the tRNA sort of did look like a T, that's where it got its name, and it was designed to basically go in the cytoplasm, grab the amino acid that was needed, and then when the ribosome called for it, basically because it's in the instructions of the mRNA, it would place it and drop it off. And this will make a little more sense in a second. So if we look at another tRNA, you'll see that there's an area that can grab on a amino acid, and there's an area called a anticodon. So remember how a codon was basically that 3-letter code that was in the mRNA that said, hey, this is the amino acid you need to get? The anticodon will be the opposite. You'll actually see a couple in a second, and that will tell us that we have a match. They sort of come together kind of like a magnet, like the positive end and negative end come together, and when they have that perfect match, they drop off that amino acid. And then this all happens in the ribosome, and the ribosome kind of has 2 parts to it, and it has a couple of pockets in it, and we'll see it in a second. So this is the mRNA. These are the 2 halves of the ribosome. It kind of looks like a hamburger bun. And then these guys are going to be the tRNA. We have 2 of them here. And one of them has just brought the amino acid. It's going to transfer it to this guy, and then this guy will float away. And you'll see that in a second. So this growing peptide chain, or growing amino acid chain, is that primary level of protein structure, and eventually will make a protein or a hormone or an enzyme, whatever we need. So let's kind of get this whole thing started and talk about the initiation, elongation, and termination. Initiation is basically where the mRNA comes in, and then we find that starting codon called AUG. From there, we're going to start to elongate, start to make that amino acid. And finally, at the stop codon, the period, we're going to go ahead and release the process, and now we know that the protein is done. Here are all the players again. Just kind of as a quick recap, replication was going from DNA to DNA. We're not doing this right now. We're just doing this process right here. We've already done the DNA going to RNA. Remember that? We converted the DNA into an RNA code, which left the nucleus, and then we did that splicing event where we just made it all exons and left the cell, the nucleus. And finally, now we're in the rough ER, and we're going to do this process. This all occurs in the ribosome. And basically what happens is, if this was the code for AUG, it would go ahead and get a nucleotide, and that nucleotide would then attach and then start to make this longer and longer and longer. So we're using the mRNA as a template to build our amino acids to complete it.